This video will show you how to solve 3-point test-cross linkage problems. So we've seen in class that if we have access to recombination data, we can make measurements as to how far apart two loci or two genes are along the length of a chromosome. We can infer from that recombination data that if two loci are far apart from one another, that it is likely that a crossover will occur between them, and if two loci are close to each other on a chromosome, that it is less likely for there to be a crossover between them. I also want to point out that if by chance the recombination frequency is greater than 50%, that what that means is even though two genes are located on the same chromosome, that the probability, the likelihood of a crossover occurring between those two loci is so high that those two genes are behaving as if they are on separate chromosomes. So even though those two loci are linked to each other, they're behaving as if they're not with such a high recombination frequency. In fact, they're going to behave as if they're on separate chromosomes and are assorting independently from one another. So if we have access to that recombination frequency data, we should be able to construct a map of the loci relative to one another along the length of a chromosome. We can express that map in terms of centimorgans or map units or even just recombination frequency. But I do want to point out that although these are all different measurements that can be used to determine map distance, the distance between two loci on a chromosome, they are not actually measurements of physical distance between two loci. They are measurements of the relative distance between two loci along the length of a chromosome. So how do you go about solving a 3-point test cross problem? Well, first, we're going to make the assumption that we're actually dealing with 3 loci, where at each locus there's a different gene that influences some aspect of an organism's phenotype. So in this example, we have an eye color locus and then 2 wing phenotype loci. Also in this problem, the sample problem, we have 2 parental stalks. One parent is homozygous wild type at the V locus, and is homozygous mutant at the other 2 loci, the CV and CT loci respectively. The other parent is homozygous mutant at the V locus, but is homozygous wild type at both the CV and CT loci. So what we're going to do is we're going to allow for a parental cross to take place. We're going to produce an F1, and then from those F1 offspring, we're going to test cross the F1 with an individual, in this case a male, who is homozygous mutant at all 3 loci, the V, CV, and CT loci. So here's the parent that we started with, or at least one of the parents that we started with. We can see the homozygous wild type individual at the V locus and homozygous mutant at the CV and CT loci. If we replicate the DNA and then allow for meiosis to run its course, we're going to produce a single combination of alleles at the end of meiosis 2. Well, here's the second parent, homozygous mutant at the V locus, homozygous wild type at the CV and CT loci respectively. Replicate the DNA, and again, we'll produce one combination of alleles at the end of meiosis 2. Now, if we used one of those sex cells from this parent and another sex cell from this parent, we could create an F1 generation from that fertilization event. So here is the progeny produced from a cross between those 2 parents' sex cells. Here we can see that this individual is heterozygous at the V locus, the CV locus, and the CT locus. If we replicate the DNA and then allow for meiosis to run its course, now we can see there are 2 unique combinations of alleles produced at the end of meiosis. Now, as I mentioned on a previous slide, we're then going to take this F1 individual and test cross. So here is the test cross. Remember that a test cross is a cross between an individual that is heterozygous, in this case, at all 3 loci, and then a homozygous mutant at those same 3 loci. Replicate the DNA, and here are our combinations of alleles produced at the end of meiosis 2. Now, in this case, we had 2 different combinations of alleles that can be produced from the first parent, 1 combination of alleles from the other parent. So what I would ask you that you do is predict both the phenotypes and the ratio of phenotypes that you would expect to appear as the results of a cross between this individual and this individual. Well, here are the actual results from that cross. Not quite what you expected, is it? Clearly, there is an outcome that we would have expected, but then there are lots of outcomes that we wouldn't have expected. So you made a prediction based on the genotypes of each of the parents that we saw in the previous slide, but what you predicted, what you expected, does not match what you observed. So let's see if we can explain what happened. Well, in order to understand what happened, we first have to determine the gene order of V, CV, and CT along the length of the chromosome. Now notice we're assuming that V, CV, and CT are all located on the same chromosome. That wasn't guaranteed. It's possible that V and CV are on the same chromosome, and CV and CT are on different chromosomes. Or maybe even that V, CV, and CT are on the same chromosome, but so far apart from each other that they're behaving as if they're on different chromosomes. Regardless, we have to figure out the gene order, and it's always a good place to start in order to assume that V, CV, and CT are all on the same chromosome. We can disprove that later. If indeed V, CV, and CT are on the same chromosome, we can replicate the DNA. And then what I want you to notice is that we're going to allow for a double crossover to occur between V and CV and CV and CT. And the result of that double crossover will be to exchange the position of the CV plus and the CV alleles. Can you see here how the CV plus allele is now on this chromatid and the CV allele is on that chromatid? What we've done is had a double break between each of those two loci. We've swapped the genetic material in between the V and the CT loci, and we've reattached or repositioned each of those alleles onto each of these two non-sister chromatids. So that's the result of the double crossover, and here are the resulting sex cells that would occur, or that would be produced at the end of meiosis II. But that certainly isn't the only possibility in terms of gene order that could have occurred. We had V, C plus, CT, but it's entirely possible that we could have a CV, V plus, CT order, or simply a CV, V, CT order, replicate the DNA, allow for a double crossover to occur once again. Again, we're going to change or alter the position of the V plus and V alleles in between the CV and CT loci, and then generate the sex cells at the end of meiosis II. There's one more possibility that we have to account for, a CV, CT, V gene order, DNA replication, double crossover, there it is, and these are the resulting sex cells. So how can we use each of these three possible gene orders and the resultant sex cells that are produced at the end of meiosis II, given each of those three gene orders, to help us figure out which of these three is the correct gene order? Well, I want to call your attention to two allelic combinations that are produced from this gene order at the end of meiosis II. Here's the first combination of alleles, CV plus, CT, and V. And here's the second combination of alleles, CV, CT plus, and V plus. I would encourage you to write down those combinations of alleles so that you can use them to understand which of these gene orders is going to be the correct one. So again, write those two combinations of alleles down on a separate piece of paper. So I hope you can figure that a double crossover is a rare event. So when you go back to this cross and you look at the results of the cross, you're going to see that two of these outcomes didn't occur very often, 3 times and 5 times respectively. Well, that's a clue that these phenotypes are the result of the double crossover. So again, keep that in mind. If you're working with a 3-point test cross and 2 of the phenotypic outcomes are the least likely, they are most likely the least likely because they are the result of a double crossover. Okay, this next part's tough, so try to follow along. I'm hoping that you can see that the most common phenotypes are the direct result of an individual inheriting a parental combination of alleles, V, CV plus, CT plus, or V plus, CV, CT. Remember how we had that situation where the parent was heterozygous, that triple heterozygous parent, where they were heterozygous at the V plus locus, heterozygous at the CV locus, and heterozygous at the CT locus? Well, here we can see what happens if there's no recombination, no crossing over between any of the loci. these fruit flies look like their parents. In particular, they look like their mom. So how can we use this information to figure out what is the correct gene order from the previous slide? Well, what I would encourage you to do is to look at these combinations and then look at these combinations. And what you're going to find is that the only way to produce these offspring in these proportions is if there's a double crossover between the CV and CT loci and the CT and CV loci. In other words, the gene order has to be CV, CT, CV. Okay? Go back to the previous slide and try to prove to yourself that that makes sense. Go through the other two combinations and show that a double crossover, if only when the gene order is in that particular configuration, remember, CV, CT, and CV would allow for those offspring to appear as a result of a double crossover between those three loci. Okay, so we're making progress. Now we know the gene order, and we know which of those offspring are the result of the double crossover. Now we're going to go ahead and actually figure out the gene distance between those loci. So you'll notice I now have the correct gene order written up here at the top of the page. I'm going to go ahead and replicate my DNA, but this time, rather than allowing for a double crossover, I'm only going to allow for a single crossover. And I'm going to allow for that crossover to occur between CV and CT and CV and CT only. So you'll see, here's the result of that single crossover. The CV plus allele is now on this chromatid. The CV allele is now on this chromatid. And here are the sex cells that would be produced as a result of meiosis. All right, we're making progress here. The next step, again, given that gene order that we discovered on the previous slide, CV, CT, V, this time, you can probably guess what's going to happen. I'm going to replicate my DNA, but allow for a crossover to occur between the CT and V loci. So I'm allowing for the possibility of a single crossover event to occur between each of these two pairs of loci taken separately. First it was CV and CT, and now it's going to be CT and V. So single crossover, and of course what I can see is that there is an error on this slide, and unfortunately it's too late in the evening for me to switch this, so hopefully you can just go along and see what I'm going to try to do here. Of course, if I were to exchange the alleles between CT and V, there shouldn't be a V plus and a V here. The V plus should be here, and the V should be there. Hopefully you can see that. Everything else on the outer chromatids will remain the same, and will produce these four combinations of alleles, except of course the V plus should be on this chromosome, and the V should be on this chromosome. Sorry about that, hope it doesn't confuse you, but I just noticed that mistake, and it would take me far too long to go back and fix that mistake at this point in time. All right, so we're back to our table. How can we use this table to help us figure out which of these offspring are the direct result of single crossovers rather than double crossovers? So here's the same cross that we had a moment ago. This time, of course, we're focusing on the single crossover and not the double crossover. And we can see that the results of that single crossover are right there. And if you go back and look at the table, what you're going to find is that the progeny that appeared at 40 times and 45 times are the direct result of this combination of alleles appearing in a particular offspring. Okay? Whether it was CV plus CTV plus or CV CT plus V, which are the direct result of a single crossover between CV and CT, we now know which of those possibilities from the previous slide are the result of a single crossover between CV and CT. Again, here's the picture that I showed you previously. Again, please excuse the fact that there should be a V plus here and a V there, but regardless, this time a single crossover between CT and V will produce these two combinations of alleles. Of course, the V plus should be here and the V should be there. But if you go back and look at that table of progeny, 89 individuals will be the result of a single crossover between CT and V. 94 individuals will be the result of the other allelic combination that is the result of that crossover. So now we have a way to know which of the individuals from that table are the result of a crossover between CV and CT, and are the result of a crossover between CT and V. Great, so now we're almost done. Now all we have to do is take those numbers that we found and use them to calculate a recombination frequency for each pair of genes. I started with CV and CT here. I certainly could have started with CT and V. It doesn't really matter. You could even focus on CT and V if you so chose. It really doesn't matter. But given the way that I've set up the problem thus far, we're going to use CV and CT first. All right, so if I want to find the recombination frequency between CV and CT, I know that the recombination frequency equals the number of recombinants divided by the number of offspring. So what are the number of recombinants in this case? Well, we saw previously that the number of recombinants due to a single crossover between CV and CT is 40 and 45. But don't forget about those double crossovers that would have also produced recombinant offspring. Remember, we saw 3 and 5 offspring respectively that were the result of the double crossover, and we're going to divide that by the total number of offspring. I won't make you do that math. It is 1,448. And when you do that calculation, you find that CV and CT are 6.4 centimorgans apart from one another. Okay? All right, now for CT and V, similar setup, percent recombination frequency. In this case, will equal 89 plus 84. Those are the results of the single crossover between CT and V. And now, don't forget about those double crossovers. They still are recombinant offspring divided by the total. And this time, your recombination frequency works out to 13.2 centimorgans. So now we have the map distance, we have the relative distance between CV and CT and V, and we can construct a very simple genetic map that shows the distance between all three loci. Hopefully you can see that the distance between CV and V would be the sum of the distance between CV and CT and V, because we know, again, the gene order is CV, CT, V. So if we know the distance between CV and CT, and the distance between CT and V, we know the distance between CV and V. All right, so that is how you want to go about solving a 3-point test cross problem. I've included several practice problems for you to work on. These are the same practice problems that were part of the homework assignment from a couple of nights ago. Hopefully you have a better chance at solving these problems now. If you need help, please come and see me. But I will leave the answers with the substitute so that you can check your answers after you have finished each problem.